Nowadays we all use sunscreen. Last year I went on this scuba diving and marine biology summer camp, and I woke up every day to the same call from the captain of our boat. They would yell, "Campers, lather on some sunscreen. We're diving in a half hour." And whenever anybody jumped in the water, I'd see like this cloud of white in the water around them, and I realized how much sunscreen each person was putting in the water. And any swim coach or scuba instructor will always strongly advise you to wear sunscreen, but sunscreen has some extremely bad side effects on the environment. And I've known for a while that sunscreen is bad for the environment, but I never realized how much people use and how bad it really is until reading this paper. So today I'm going to present on this paper that describes the effects of BP3 on coral planula. So the BP3 is short for Zone 3, which is a scientific name for oxybenzone, which is this chemical in sunscreen. So sunscreens overall are bad for the environment, but this paper mainly talks about BP3, and coral planula are the coral larvae, or like the babies before they settle. So why should you care? Of course, you all as marine biologists know why coral reefs are so important, but a lot of people probably just think that corals are just pretty rocks in the ocean. But these coral reefs are actually some of the most important ecosystems on this planet, and they are a source of food for marine life and, of course, humans, because we rely on these fish. And they're a shelter for sharks, sponges, jellyfish, octopi, manta rays, and actually 4,000 species of fish, which I thought was pretty amazing. And since corals are these massive structures in the sea, so they actually end up stopping some big waves from hitting our coastlines, which is really important, especially with the rising sea levels. And due to tourism and also fisheries, coral reefs are worth $375 billion of the world economy yearly, which I thought was pretty amazing. And but it's really sad because over 50% of coral reefs on Earth have died in the past 30 years, like just 30 years. Scientists are predicting that 90% will die in the next century. And I thought that was pretty shocking and really sad. And this picture is just the Great Barrier Reef a decade back and now. And you can see it's really bleached. So just some background information. Wait, sorry, what was that?
Yeah, there's a lot of old dead coral there. So all those corals you see are dead. They've bleached and died.
Yeah. Oh yeah, I forgot to mention, if you guys have any questions, please feel free to interrupt. So some background information about BP3. So BP3 or oxybenzone is used all around the place. So it's not just in sunscreen. It's found in all types of cosmetic products and a lot of household products as well, like soaps. So it's not just in sunscreen. And something scary I found is that it's absorbed by your skin. So this graph shows the FDA threshold for like how much chemical should be absorbed into your skin. So the FDA says that none of it should go in the skin, but all of these chemicals here in the graph are parts of sunscreen. They're chemicals in sunscreen. Is the worst by a far shot. So studies have oxybenzone is people's blood is pretty crazy.
Hey, you're
and
kind
of
of
course,
breaking out. We've got all these contaminants in sunscreen that are getting into our skin, and they're above what the FDA deems as good or okay.
Yeah, and of course, oxybenzone is also lethal to coral. I'll talk a lot about that in this presentation. And a recurring theme in this paper that I'm presenting about is that BP3 is a phototoxicant, meaning that its harmful effects are accelerated and worsened by light exposure. So some adverse effects of BP3 that were talked about in the paper: in mammals, it increased estrogen and decreased androgen. So of course, when it's messing with your hormones, it's going to have a lot of bad side effects. And just some of them are that it causes risk to breastfed newborns, and it also causes menstrual irregularity, which is not surprising considering it's increasing the estrogen. And it's also pro-carcinogenic, meaning that it causes cancer. And in fish, long-term exposure to BP3 decreases the population overall. So it decreases egg production and it decreases the egg hatchings. So like fish, if you guys have seen it, they lay thousands of eggs. So it's reducing that number and reducing the amount that hatch out of those eggs. And it also increases gender shifts from male to female. So if there are less male fish to mate with female fish, then the population is obviously going to go down. So just some BP3 usage. These are two very, very popular sunscreen brands. Almost anybody will have them, Banana Boat and Coppertone. And here on the left, the Banana Boat has 5% oxybenzone. And the image on the right is a little bit harder to see, but it's got 6% oxybenzone. So a little bit about my beads here. I'm not going to talk too much about the beads because I'm going to talk about them more at the end. I'll just give like a overview. So so when I learned that oxybenzone and sunscreens were so bad for the environment, I felt really bad and I wanted to do something to fix it. So I developed these beads that are about the size of poppy seeds and they can absorb sunscreens. So like if you drop them in water with sunscreen mixed in, the sunscreen will come and stick to my beads. And when you remove the beads, the sunscreen will come out too. So the beads that I created were magnetic nanoparticle beads, and I'll explain what that means. So I made the magnetic nanoparticle solution using ferrous and ferric chloride, and I mixed them with ammonium hydroxide. So this video here is just like a short clip of my beads being made. I actually made this small machine that would automate the bead making process, and it made my life a lot easier. But yeah, and the picture at the bottom is my beads after the final stage, which was magnetic decantation. So this is like the final solution. So I thought that instead of talking specifically about how I made my beads, it would be better to talk about why I made them the way they were made. So why were they magnetic? Well, if you have magnetic beads, it's really easy to extract them from the water. So if you dump them in a bucket of water with sunscreen, you can pull them out in like a second just using a magnet, which was really simple and very convenient. And the reason I use nanoparticle beads is because a bunch of tiny, tiny particles attached to each other, then it's going to be a lot more porous. So if you have like a more porous surface, that means that the surface area will be greater. Is that sunscreen sticks to these beads. So if the surface area is higher, then more sunscreen sticks to them as well. The second part of my beads is the chitosan. So these are actually chitosan coated magnetic nanoparticle beads. It sounds like a really long title, but it's not. But I'm going to explain it here. So I coated the magnetic nanoparticles in chitosan for a couple of reasons. So first of all, chitosan is widely available and it's biodegradable. So it's actually made from chitin, which is found in the exoskeleton of crustaceans like shrimp and crabs. So since it comes from these marine animals, it's native to the ocean ecosystem and it's completely sea safe. So I'm not actually going to release these beads into the ocean, but just for the short amount of time that they'll be in the water, it's sea safe. And another thing about chitosan is that it forms long polymer chains. So since it's such a long molecule, it likes to it will wrap around the magnetic nanoparticle really easily. So it's very easy to coat them. Another reason that I use chitosan, the main reason actually, is because of its adsorption properties. So once I coat the magnetic nanoparticles with this chitosan, it'll absorb this sunscreen. So if you look at this picture here, it compares the chemical structures of chitosan on the right and oxybenzone, and both have this hydroxyl ion, or this hydroxyl bond here. It's neutrally charged. Sorry, it's not an ion. Hydroxyl molecules are actually polar. So since they're both polar, they can stick together and bond really strong. And also, chitosan is neutrally charged. Oxybenzone is charged. So neutrally charged substances attract. So that helps their sticking property there. So So right now, after I put the chitosan in, this was still like a liquid solution. So if you remember the video I showed you, it was steering the magnetic nanoparticle solution. So like, how did I convert this liquid into the actual beads? I dropped them with a pipette into petri dishes with diluted sodium hydroxide, and then they formed into beads.
It's
So
cool.
this, yeah. Oh yeah, do you guys have any questions? I know I just dumped a lot of information on you. Was it all clear?
Yeah, it was really clear.
Okay, good. So this core here, can you guys see the pen that I'm drawing with? Okay, so this core here is a magnetic nanoparticle. You can see, and here is the chitosan. So it's coating the magnetic nanoparticle. And not only does the chitosan coat the particle, but it also has this bond here, and it also can absorb the oxybenzone here. Okay, now I'm going to start summarizing the paper. A little bit about their experimentation methods. So they tested 7 coral, coral species. Of course, one isn't enough. And the one that they focused on in this study was Stylophora piscillata, but they also got data for the other ones. And Stylophora piscillata is like mostly found near India and China and Australia. So they said that it may not be an accurate representation for Hawaii or the Caribbean. So I also included a data table with the data for the other reefs as well, so you can get like an accurate interpretation of reefs around the world and not just in this area. So yeah, this is a sad coral reef here. I thought this picture was pretty cool. But what they measured in the experiment was histopathology, which is like changes in tissue and tissue degradation. So there was a lot of this that they studied, and they got some really interesting results here. And they also measured cellular pathology, planula morphology, and bleaching, and planula mortality. And this is another main part here, DNA AP. So DNA AP is short for DNA abasic sites, and these abasic sites are sites in the DNA that have no base pair attached. So the base pairs are like the building blocks of DNA. So without this base pair, they're just mutations in the DNA, and they affect everything else. Like they lower their reproduction rates, and it's just very bad overall. So according to my calculations, we're screwed. The corals are so important. They're worth $375 billion yearly, and we are messing with them so bad. And the BP3 is really affecting their growth and everything, which you will see very soon. Oh, and also something that I found really shocking is that between 6,000 and 14,000 tons of sunscreen are released into reef areas each year. And this puts almost half of coastal reefs at risk. So 40%. And the reason that more coastal reefs are at risk is because people mostly use sunscreen in beaches, and it affects the reefs closer to the beaches.
Is that the total mass of the sunscreen, or is that the mass of the contaminants?
That's the mass of the sunscreen. And out of those sunscreens, most of them have 1 to 10% oxybenzone. So about the method section, I'm going to be talking about all of these in order. So first, we have culture cells versus planula. So this paper is a study on BP3's effects on coral planula, but unfortunately, planula are actually really difficult to collect. So first of all, they need to find corals that are reproductively viable. Of course, if you need the larvae or like the babies, you need the parents to be reproducing. And corals also only spawn once a year, so they release their gametes all on one night. So you need to go to the coral on the right date in the right moon phase to catch that planula. So instead of using the planula, they used cells like adult culture cells that they grew. So of course, the adult cells will have different reaction to sunscreen exposure than the planula will because they're more mature. So they made these formulas here to convert the mortality of planula to the mortality of cells and vice versa. So you'll notice here that there are 2 formulas, one for light and one for dark. And remember, that's because oxybenzone is a phototoxicant, meaning that its effects are worsened by light. So they need to have a different formula for converting for light and dark.
And this is something
So
that they tested and made, right?
yeah, yeah, I'm getting there. So in order to like test these formulas, in order to make the formulas, they needed planula in the first planula mortality rates, then the cell mortality rates, and then they were like, okay, so this is the formula. So for the Planulae collection, positively buoyant planula traps. So positively buoyant means it's going to stay upright in the water. And at the base of the trap here, they have these rubber straps that they'll on, like on the colony, so it can't just drift off. So they have these nets here, so when the coral colonies spawn their gametes, they'll fertilize, and then the planula will be trapped in the net. So they didn't need many planula, but they needed enough to make the formula of conversion. Number 2, artificial seawater creation. So W for short. When they're not use normal ocean water, that's because ocean water already has a little bit of BP3 in it. If they were to add their concentrations of BP3 to the ocean water, that would not be including the amount that's already in there. So that would be skewed. And they had to make their own artificial seawater. So for the treatment water, the salinity was 38 parts per thousand, and 22 degrees Celsius was the temperature. So they were mimicking the Israel water, which is where they were conducting the study. So that made sense. But for some reason, the water that they maintained the coral cells in was a different salinity and temperature. So I'm not sure why they did this, but it seemed a little bit weird that the artificial seawater was different in both of these. And the light that they used, remember they did the experiments in light and dark. They used 295 micromoles per meter squared per second of photon flux density. So that sounds, you don't really need to know too much about that, just that the surface gets 2000, and as you go deeper into the ocean, most of the light gets absorbed. So I think they could have gone as low as 200 here, but they chose 295. So here you can see that they had 6 different concentrations of BP3. So what they would do is they would add
I think you're being hacked. He's being hacked. The sunscreen companies are onto him.
Yeah, they're gonna steal my presentation.
Ah, there you go. That's better.
Okay. So here you can see that they had 6 different concentrations of BP3, and they would mix these different concentrations into the artificial seawater samples they made, and they would test the planula with exposure to each one of these concentrations and see how much worse the effects got across this spectrum. And another thing is that they did planula testing for 8 hours in light, 8 hours in dark, 24 hours in light, and 24 hours in dark. And each one of those had all 6 of these concentrations. So they did a lot of tests there.
Did they have
And
controls,
if you've ever seen
Sriram?
sorry, what was that?
Did they have controls?
Yeah, they had a control group, which you'll see in the pictures. Like you'll see the unharmed planula versus the harmed planula.
Did the controls get the dimethyl sulfoxide?
Yes. What they did was they put this solvent in the 5 microliters per liter of dimethyl sulfoxide, which is a solvent, in this BP3 and artificial seawater sample to mix it much better. And they did a lot of tests where they tried the test with the dimethyl sulfoxide and without the dimethyl sulfoxide. So they found out that it was not different. Like they had the same reaction to both solutions.
Okay, thank you.
So they basically concluded that it didn't matter if they added the solvent in. And you're probably wondering why 228 and 22.8 seems like really random numbers. And I was thinking that too. And they didn't say this in the study, so this is just a hypothesis. But I'm thinking it may be because the molecular weight of BP3 is 228 grams per mole. So I'm not sure why that would make a difference, but maybe they just thought that the equations would be easier if it's the same molecular weight. Now about chlorophyll fluorescence. So they use this as an estimate of bleaching, and chlorophyll fluorescence basically tells you how good your organism is at photosynthesis. So if a coral is good at photosynthesis, that means it still has its zooxanthellae, because the zooxanthellae are the ones that photosynthesize for the coral reef. And if the photosynthesis is low, that means that the coral will probably be bleached. And they use a molecular dynamics microplate fluorometer for this testing, and that's the drab little machine you see to the right. It's like just a brick. It's pretty lame. But the excitation was 445 nanometers, which is blue light, and the emission wavelength was 685.
Okay, so
Should I repeat?
excitation and emission is what it's measuring. So it shines 445 at a coral cell, and then it measures at 685, right?
Yeah.
And
So the way
it's a
that chlorophyll
proxy for how
fluorescence.
much chlorophyll is in there. Is that correct?
So for chlorophyll fluorescence, they shine blue light, and based on how much red light is reflected, they can tell you how good the organism is at photosynthesis.
It's not reflection, right?
Oh yeah, it's like how
Fluorescence.
much it gives off. Yeah,
It's
fluorescence.
fluorescence. So when light hits the chlorophyll cell, it actually makes a new light that's red, and they measure that.
Yeah.
We just need to say those things because others, even in college, might not know that.
Okay. Okay, now about DNA at basic sites. So just a small recap. The DNA AP sites are the sites without the base pair attached. So they cause lots of problems and mutations and problems with reproduction as well. So they use this Dohindo DNA damage quantification kit for counting this. And this is a colorimetric analysis. And it counts the number of the basic sites in DNA samples. And one thing here is that they only analyze the intact planula, even if the planula were counted as dead, which is why they couldn't do DNA AP site analysis for the sample that was exposed to 228 parts per million of oxybenzone in light, because all the planula were dead. Okay, transmission electron microscopy, or TEM for short. This was a really important part of the paper, and you'll see it a lot more. So at the top here, you have this electron gun, which shoots a ray of electrons, and then you have this condenser. So if you look here closely, the condenser turns the wide ray into a much narrower, focused, like laser-like blast there, and you have the specimen right there. And through all these lenses, this accelerated beam of electron will create a projection at the bottom on the fluorescent screen. So using transmission electron microscopy, you can basically study these really tiny specimens very closely, and you can see everything really with a lot of clarity. And they use this for tissue and cell deterioration analysis. And you will see a lot of pictures that they took with TEM throughout the coral mortality. I thought it was really interesting how they did this actually. They use this chemical called trypan blue, and the trypan blues, its feature is that it colors dead or damaged cells blue. And they use this machine called a Newbar hemocytometer. And what that machine does is that visually, like under a microscope, it will count dead cells versus alive cells. So together, these work together, and the Newbar hemocytometer will make a grid and then count the cells using that grid. And all the trypan blue cells, the dead cells, will all be colored blue. So that way it can count dead cells versus alive cells. And I just thought this was really interesting. Okay, this was one of the most important parts of the studies and it even aided my research a lot. And this is how they measured the BP3 concentration in ocean water. So I'll start by explaining gas and liquid chromatography. Both of those are methods that use either gas or liquid to separate the components of a chemical solution. And then mass spectrometry can be used to calculate the exact molecular weight of each of those components that were separated. So LCMS and GCMS are the gas chromatography and mass spectrometry, and the same with the liquid chromatography. They're all combined together, the methods, and it uses it to basically separate the seawater and then measure the weight of the oxybenzone in there. And the machine they used for the liquid chromatography was the Spark Holland symbiosis, and the machine on the right is what they use for mass spectrometry. So when collecting these samples of ocean water to find the BP3 concentrations in them, the workers who collected these samples were decontaminated with an Alkanox liquinox, which is a concentrated detergent, and they also didn't use any non-organic personal care products for 21 days before collecting. And I thought this was really cool, right? Like imagine whenever going into your lab, you have to spray yourself down with detergent. This like I just admired their procedure. It seems really detailed, really careful.
Yeah, I don't do that.
Yeah, yeah, yeah. I don't know how necessary it is, but it seems pretty cool. So the last part of measuring the BP3 concentration, they measured it 100. They took 100 to 500 milliliter samples, 35 centimeters below the surface, and served it frozen. Stored it frozen. But the main thing here is that they used these Phenomenex C18 solid phase extraction columns, and these are really cool. Um, these are really cool things. I really wish I had one. So when you like get some solution into these extractions, it holds the target analyte here, or in this diagram, the blue diamonds, and it will shoot everything out, like the red contaminants and the water. So they didn't. They said that they used these, but they didn't actually say what the target analyte and the contaminant were. But I'm guessing that they were trying to get rid of other pollutants in the water other than BP3, so the mass spectrometer would not get confused by measuring any other pollutants. But I wish they were a little bit more clear here. So now for the results, the first thing I want to talk about is change in morphology. I know this slide looks super disgusting, but don't worry, you'll see why. So in the control, the unharmed planula, you'll see that their morphology is in the shape of a cucumber, right? It's like long and thin and looks super cool. And it has these rows of zooxanthellae. So all these rows here are actually gastrodermal cells with zooxanthellae on them. And it's also transparent, so you don't see any opacity here. But then BP3 comes along and totally ruins these chloroplanula and makes them look super ugly. So at these different concentrations of light, you'll see that, of BP3 with light, you'll see that this cucumber morphology turns into a dewdrop. So it becomes much more rounded, and you can see that just like compare figure A and E, it's such a drastic difference. Looks really deformed. And also the rows of zooxanthellae here fade. And I'll explain why in a second. So you see here, you can barely see the zooxanthellae in figure C, and you can't see them at all in figure E. So this happens because of this thing called symbiophagy. And this is basically when at the end of a symbiotic relationship, one of the symbiotes actually ends up digesting the other one. I know, super violent. But when zooxanthellae are exposed to this BP3, they actually start hurting the coral, and in return, the gastrodermal cells in the coral, or the digestive cells, actually digest the zooxanthellae. So yeah,
Damn.
be careful. Be careful who you make friends with. And another thing is that this opacity you see here in figure A, it's very transparent, and in figure E, you can barely even see it at all. It's much more opaque. And the reason that this happens is because of ossification. So ossification is bone development, and BP3 greatly accelerates that. So it accelerates it to an extent where the planula is actually enveloped in its own bone, which sounds super creepy, and that's why it's so opaque here. And also in the control, the planula, its oral pore or its mount actually expanded 3 to 5 times in diameter, but after BP3 exposure, the mouth ended up shrinking back into the body, just recessed. Some more Change in morphology. So here's the cilia. So cilia are these tiny, tiny hairs that are found in a lot of organisms, including humans. So they're actually in our noses and they're there to trap dirt when you breathe. And in coral, they're used for kind of similar stuff. They're used to get rid of tiny sediments in the coral. So they have a pretty important role here. After 4 hours, there was a major reduction in cilia movement. And after 8 hours, the planula just stopped moving. So because of these cilia, the planula were in constant motion. They were being propelled around a little bit, but they stopped moving after 8 hours of BP3 exposure. In figure A on the left, you'll see all these like needle-looking things on the side poking out from this line. And those are the cilia, the tiny hairs, but on the figure on the right, you'll see nothing. Which is just completely blank. It's pretty scary. There are no cilia at all. So now I'm going to talk a little bit about how the cell damage went within the cell itself. So after 228 parts per billion exposure for 8 hours in light, there was effects in the epidermis, the gastrodermis, and the mesoglea. So the epidermis is the outermost layer of the cell, and then the gastrodermis is on the inside. Again, it's the digestive part, and the mesoglea is this like jelly-like layer that separates the two of those. So there was severe tissue degradation or severe tissue lysis on the epidermis, especially, and there was cellular degradation. And in fact, the degradation was so bad that the mesoglea actually had abundant debris, which was pretty scary. And the greatest deterioration was on the surface of the epidermis, and especially with the cilia, or no cilia at all. And in the middle of the epidermis, there was this thing called autophagy. So autophagy is the cell's mechanism to deal with damaged cells. So if it detects that there are a lot of damaged cells in an area, it will these things called autophagosomes will clear out all those damaged cells. So this way, so increased autophagy means that there's increased cell damage, because why would the cell need to clear out damaged cells if there were none? So just showing that there were a lot more cell damage here. And another effect was that there was delamination of the nuclear bilayer membrane. So delamination is just another word for fracturing. And here in this diagram, we see the nuclear envelope is just another word for nuclear bilayer membrane. And that's this like purple layer surrounding the nucleus. So the bilayer membrane is really, really important. It's this selectively permeable membrane. So it lets in certain things and it sends certain things out. So it's really important because that's what protects the nucleus of the cell, which is responsible for everything else. So if this bilayer is fractured, that's obviously going to cause a lot of problems for the cell. So gastrodermal damage, again, gastrodermal, that's responsible for digesting, and it faced extensive trauma, autophagy, and loss and vacuolization of zooxanthellae. I'm going to use a pen again here. So autophagy, again, is clearing out damaged cells. So if there was extensive autophagy, it means there was extensive damage to the cells. And here in the diagrams, you'll see this Y. That's the yolk of the gastrodermis, and you see that same yolk here. Oh, and by the way, each of these pictures, the E to F to G, are the progression of cells that were facing higher and higher levels of BP3. So E is a control, and F is a high BP3 concentration, and G is the highest level of BP3 concentration. So in E, you'll see that around the yolk, there's not many dots, but here, external to the Y, the yolk, there are all these black dots, right? So that's all the vacuoles that are formed. Oh yeah, so vacuolization is just the formation of all these extra vacuoles that a cell doesn't need, and it indicates cell damage. And here in this figure, you see ZX, that's zooxanthellae. You see like only a couple of vacuoles here, but in G, when it faced BP3 exposure, so the ZX, these are both zooxanthellae. You can see both of them have faced really heavy vacuolization. So that means that it had a lot of cell damage there. Okay, Zooxanthellae morphology. So of course, zooxanthellae are really important to coral reefs. They give them 90% of their energy through photosynthesis. So if the zooxanthellae are being deformed, that's not good at all. So there was a lot of chloroplast degradation. So chloroplasts are the thing in algae that are responsible for photosynthesis. They're they're parts of cells. So if there's the chloroplasts are ruined, that means the photosynthesis is going to go down. And this especially happened in thylakoids, which I will talk a little bit more about in the next slide. And there was also a homogenization of chromatin. So chromatin is stuff that has DNA and RNA. Really important. It contains your genetic material. And levels of chromatin: heterochromatin, which is really densely packed, and euchromatin, which is lightly packed with chromatin. So homogenization means that these both were made into like one medium packed chromatin thingy. Sorry, that was very badly phrased, but the point is the chromatin the, the chromatin concentration throughout the cell. About the thylakoids, thylakoids are they contain chlorophyll and they're parts of the chloroplasts. So thylakoids contain chlorophyll, which is the thing in chloroplasts that absorbs the light for photosynthesis. And thylakoid lamellae are just stacks of these thylakoids. So thylakoids are like really thin discs. So the lamellae are the stacks. And these thylakoid stacks were very diffuse, meaning they were really far apart. So if you look at this diagram, all these white spots, the lamellae are really spread apart. And also BP3 causes photooxidative stressed thylakoids. And photooxidative means that it degrades it in presence of oxygen along with UV or artificial light. This is coming back to the theme of like phototoxicant. It's worsened by light. This is photooxidative. So when it comes in contact with air and light, it causes damage to these thylakoids. So effects in darkness versus light. In darkness, the effects were not as far felt. But remember, this is all subjective. The effects were still really bad, enough to kill the planula, but not as bad as they were in light. There was less vacuolization, so less vacuoles were formed, and less delamination as well, which is, again, the fracturing of the nuclear membrane. So overall, slightly healthier planula, but not really. And also the degradation stopped at the gastrodermis. So it went through the epidermis and to the mesoglea, but it didn't go any farther than that. So the digestive part was not actually damaged, except for 228 parts per million. At the highest concentration of BP3, everything was damaged, even in darkness. So there's this thing called an extracellular matrix, which is a three-dimensional network that provides like support to surrounding cells. And there's an extracellular matrix between the epidermis and the mesoglea, and another one between the mesoglea and the gastrodermis, so in each level. And in darkness, only the outer extracellular matrix was damaged, so the one between the outer layer and the mesoglea, but it didn't go any farther than that. Okay, so this is the meat of the presentation, the LC50, EC50, and EC20 values. So you guys have probably heard of LD50, actually. This paper used some weird terminology and for some reason called LC50, but it's the same thing. It's just a lethal concentration. So LC50 is the concentration of a chemical that's required to kill 50% of the organisms. So in this case, 50% of the EC50 is the amount needed to affect 50%, and EC20 is lower than that, the amount needed to affect 20%. So they actually had to estimate all these values. Like they didn't actually try all of these LC50, EC50, and EC20 values. They estimated using all these statistical analyses here because it would be too hard to actually try out all of them. Their main two methods were regression models, which is just a straight line. They basically thought, okay, this concentration causes, and then this higher concentration causes this amount of damage. So they could graph that. and basically find where the LC50 and EC50 values were. And for the regression models, the coefficient of determination were between 0.91 and 0.97. So the closer this value is to 1, the better. So this coefficient determination was really high. So this model was a really good fit for the graph. And they also used all these other statistical analyses, which I'm not going to go into detail right now because they're not too important. But one thing that I'd like to know is the difference between the predicted value and the observed value were randomly distributed, meaning that like concentration went up, the inconsistency did not. So it was just randomly distributed throughout. And another thing that was that really bothered me here is that I think there were some typos on this graph. If you look here, the LC50 for 8 hours in light is actually, for some reason, lower than the EC50 for 8 hours in light. So think about that for a minute. The amount needed to affect the planula is more than the amount needed to kill the planula. That makes no sense, right? It should, of course, be the other way around. So I think they meant micrograms per liter instead of milligrams per liter here, but I'm not sure. So I don't know why they didn't
prove. Because sometimes an effect that you're measuring, like maybe the cell never makes it to that level because it dies before it can like actually have the effect. Does that make sense? What if you know what I'm saying?
Yeah, that makes sense, but it still shouldn't be higher, right? It should be the same level there.
It takes a little bit of thought, that's for sure. And it's especially weird that, well, for the 24-hour light period, it's better, right? The 24-hour period, it seems more reasonable. Is that right?
Yeah. But yeah, they had a couple of typos throughout this paper, which was just an annoyance. But yeah, this data table was actually really important for my research too. So a little bit more about these values in DNA at basic sites. So they measured this across the concentration gradient. So if you remember, they measured BP3 exposure in 6 concentrations of BP3. And in light, the amount of DNA at basic sites for the control went up by 10-fold for the highest concentration of BP3, which is a pretty insane amount. But in darkness, it actually increased a 20-fold from the control to the highest value, which was, yeah, really scary.
And fold just means like times, right? So like 10-fold is 10 times more. Yeah. 20-fold is 20
yeah,
times
like here
more.
It's about 40 and it goes to like 400. So it's about 10-fold, and here it's about 20 and it goes to about 200. So that's a
what does that say for the 0 concentration then? Just by being in light, you're getting DNA AP sites. Is that right?
Yeah.
So DNA AP sites, it's common. All cells experience DNA damage, and that can just be from light or chemicals in your system or just doing exercise. And cells have a way of actually fixing that damage, right? But then when you increase the pollutant concentration, you see that damage is increasing a lot. Yeah.
And this is going to cause all types of problems like mutations and reproductive issues, or like the DNA reproduction. And different species, of course, have different levels of sensitivities. So these are the 7 species that they experimented with. And you'll notice that P. astroides is the most resistant. Sorry, forgive my pronunciation. So it's the most resistant, except for the Stylophora piscillata in the dark. So another thing that kind of bugged me about this table is that they did light and dark for only one of the corals, but not for the other ones.
Were the other ones in the light?
That I'm not sure, they didn't say, but I think they were in the light, because they said that P. astroides is the most resistant, and it has to be in the light, because if it was in the dark, then that would mean it's less resistant than Stylophora piscillata.
Right.
So, but that's just my assumption, because they didn't actually say in the study. And the reason you see confidence intervals here is because these are all statistically estimated. They didn't actually try them all out. So environmental analysis, I mentioned how they measure BP3 concentrations in the water, but I didn't talk about where. So they took samples from like over the world. So 3 samples from the USVI and 3 samples from Hawaii. And they also found, on a side note, that BP3 contamination can actually travel greater than 0.6 kilometers, which is crazy. I mean, it can affect corals that are half a kilometer away, which is pretty dangerous. And I made a table of the different sites they tested it and the different concentrations they found. So if you look at Caneel Bay here in the USVI, it only had 17 visitors on the sampling day. Not a very crowded place, and you'll see that there was no BP3 detectable. But in contrast, if you look at Trunk Bay, they get 2,000 up to 2,000 visitors daily, and there were 180 swimmers and 130 sunbathers near the place they measured at the time. And they measured next to a large coral and east of the coral. So they measured in 2 sites there, and they found really, really high concentrations of BP3. So 1.395 milligrams over liter and 580 micrograms. So if you go back to the data table here, you'll see that this is enough for the LC50 for 24 hours in light, meaning that if a coral was in this water for 24 hours for just one day, if it was in this light, it would kill 50% of the planula. And most of these concentrations are enough for the EC20s and EC50s. So if a coral was in any of these places for 8 hours or 24 hours, they would start feeling effects, which is really impactful for me,
Yeah,
actually. That
specifically bad
the
for the
coral,
acroporas, right? They were pretty sensitive.
yeah, yeah. Oh yeah. So in fringing reefs, like the acroporas species are the first to go locally extinct, and um they're the most affected by this. And in boulder corals, like the big boulder corals, they're the most resistant, and they can actually face exposure, well, like on and off exposure. They can't really face it healthily, but they can kind of face it for up to a decade, which is pretty cool. But and also another thing is that here, the the a the P. asteroidus is the most resistant, and this coral is 4.5 times as resistant as the second most resistant coral that they studied, and 38 times more resistant than the weakest coral they studied. So it's a very hardy species. Is something that was interesting. So, as if this wasn't enough, it gets worse. There are 2 main types of corals, and one is brooded coral. So the brooded corals, they fertilize and they release the planula ready to settle. But the one that's also really common is broadcasting corals. So these release gametes like eggs and sperms into the water, and then they have to fertilize in there. So once they fertilize these broadcasting planula, they actually drift around on the ocean surface for a couple of days. Period. Planktonic means that they're carried around by tides and currents, and they can't swim well enough to fight these forces. And I don't know if you guys remember, but near the beginning of the presentation, I said that these corals are facing the light at the surface of the ocean is 2,000 micromoles per meter squared per second. So in this study, they only exposed them to 295. But considering that these planula actually drift on the surface for 2 to 4 days, they're going to face 2,000, which is way higher than the 300 that they gave in the study. So the phototoxicant and photooxidative effects of BP3 are going to be far worsened by this like major increase, right, in light.
Well, it could be
So
far
these,
worsened, right? But they didn't study that.
yeah, could be. And even without BP3 effects, actually, like remember, just being in light increase the DNA at basic sites, but that's common. So some of the demographic effects, So when these planula are affected, it's not just like if they survive, they're good for life. That's not how it works. So if a planula is damaged, there's issues in the settling and juvenile survival and reproduction. So like growing into adult life and its entire adult life, it's going to face um, challenges. So like these corals, suppose they were exposed to BP3 exposure as planula, the study said that they'd be unfit to face challenges like climate change later on. So even if they do somehow survive this BP3 exposure, they're going to have these complications later in life. And also another thing in coral is that when there's a germ or disease, when there's BP3 damage in specific, these cells do not isolate. They actually reproduce, cells replicate. So even a little bit of damage will be far worsened. So that's basically done. I'm done basically with the summary of the paper, and I'm going to be talking a little bit more about my beads now. Remember, my beads, they absorb sunscreen. So if you put them in a water with sunscreen, the sunscreen will stick to them, and when you pull them out, the sunscreen will come out as well. I'm going to be talking about the results. My first stage of analysis was visual analysis. So if you look at this figure in the middle, you'll see these are my beads, and you'll see like this coating, like this gloss around them, and that is actually oil or the sunscreen. And it's kind of hard to see here. So if you look at them under the microscope, this black part is all my bead, and this yellow stuff that you see around, the yellow coating, is all the sunscreen. So clearly a lot of
Chitosan?
Sorry?
That's not the Chitosan?
No, that's the
Oh, interesting.
sunscreen.
And did you do this in a container or something? How did you get the sunscreen from some source and onto your beads?
Okay, so I had this bucket full of, so this is my very preliminary analysis, so I didn't record the data too well, but in a bucket of seawater, I put 10 ml of sunscreen and mix it in really well, and then I dropped my beads for 30 minutes, and then I pulled them out, and then they ended up looking like this. And yeah, the chitosan, you can't actually see the chitosan because there's so much little of it, and it just spreads over the bead. So this is actually the oil, the sunscreen.
Wow.
Mm-hmm. And here's some more visual observations. So these Saturn-looking specimens here are actually my beads with the rings of sunscreen around them. And here too, you can see all the sunscreen that was sticking. So now for the main analysis, I use this thing called the spectrophotometer. The spectrophotometer uses light analysis to tell you how much of a certain substance is in a solution. So you'll enter its wavelength, and it will shoot light of that wavelength through the cuvette with the substance. And based on how much light it receives on the other end of that beam, it'll say, okay, so this much light was absorbed. With the spectrophotometer, you enter a blank reading. So you just give water with no sunscreen, and then you give water with sunscreen. So there's always a control. The thing with the spectrophotometer, I use in absorbance. So how much your solution absorbed, how much light, but it gives them in this decimal points, like 0.04 or 0.05. But those values on itself don't mean much to me. So I use this thing called Beer-Lambert's law. But do you guys have any questions so far on like how the photo spectrometer works?
What's on your x-axis?
Oh yeah, that's what I'm about to explain. So Beer-Lambert's law basically says that the relationship between the concentration of a certain substance and the absorbance that the photospectrometer gives is going to be a linear relationship, so a regression line. So on the x-axis here, I have the concentration of sunscreen that I put in these samples of seawater. So like it's in grams per 15 ml. I used for my beads analysis 0.12 grams in 15 ml of water. So I know this is a pretty high amount, but this was just like my first to see some kind of readings come here. And then for the concentrations for, let's say 0.02 grams per 15 ml, I found that this first purple dot, it had this absorbance. And then I kept mapping these concentrations to these absorbance values.
Your microphone is cutting out again.
I'm definitely
really like
being
straight
hacked.
line though. That's awesome. good
Yeah, I
relationship.
know. You won't believe how shocked I was when I saw this, because all I've ever faced with the spectrophotometer was failure until I discovered the Beer-Lambert's law, and it just solved all my problems.
That's
So
great.
yeah, it maps the concentrations to the absorbance, and using this line of best fit, I can I can start experimenting with my beads. So the one I used for the first experiment was I put 0.12 grams per 15 ml of sunscreen in the seawater, and it gave me this absorbance value. And then I put in 0.174 grams of my beads in that solution, left it in there for 30 minutes, and took it out. And then I put that same solution back at the spectrophotometer, and it gave me this absorbance. So then I mapped this Y value on the best fit line, and I found the X value, and I found what concentration, or like how much sunscreen it removed from the water. And I found that it actually absorbed 38% of the sunscreen, which for the first test, I thought it was pretty encouraging, pretty significant. And I tried the same test this time with 0.332 grams of beads, and it actually absorbed 72% of the sunscreen in the original control.
Wow, so it's like a doubling.
hmm
That's great. Yeah.
Yeah, so when I pulled the the amount it pulled out. So did the whole Beer-Lambert's law make sense, and how I found out the concentration it pulled out?
Yeah, that was awesome. You're getting a lot of head nods. I don't know if you see us all at the same time as your
I don't
presentation.
see any of you.
Yeah, yeah. They're
Sorry.
all nodding.
Yeah.
They're smiling and nodding.
Okay. Um, so yeah, this is all just like a first level analysis, but it was a pretty big step. So I could actually quantify how well my beads work, but I still needed to do a couple of things, which I'll talk about in the next slide. So the reason I was so interested in this paper is because until I read this, I just had like an idea of my beads, like, okay, it just absorbs sunscreen, it absorbs this much. But that by itself means nothing, right? I need to actually make this a practical quantification. So before, I just knew that in a concentration of 0.12 grams over 15 ml of sunscreen, I needed 0.3 grams of my beads to absorb 78% of sunscreen. But what does that actually mean? So using what I learned from this paper, I can mix a concentration of sunscreen that is naturally found in oceans, and they had a list of concentrations of BP3 found in water in different areas of the world. So using the number of visitors on a beach, I can predict the amount of sunscreen in the water, and I can even measure it using the spectrophotometer. And I can mix a sample of that concentration and then put my beads in it. So it'll be like putting it in real ocean water. And this is obviously much more meaningful than putting some random concentration. And another upside of doing this study is that I also know what concentration is lethal to coral reefs and what concentration harms coral reefs. So I can put my beads in the sample that's similar to ocean water, and I can actually see if my beads could reduce the concentration below the LC50 level or below the EC50 level. And if it did, then I can actually claim that, okay, my beads are useful. You need this many grams of my beads to make an effect and to reduce below the lethal concentration. So once I do all of this further analysis, then I can actually claim that my beads are good and where I can use them, and I can do some more research on like where I should use them, or like what beaches I could use them in. And another thing that I learned from this study was the statistical analyses. So I didn't actually understand too much of these, but I at least know like what they used. So in the future, if I ever want to do an in-depth analysis like they did, I can use the same analyses. And I also learned about precision work. So I spared you guys some of the boring details of this paper, how they measured everything, but
Thank
there's
you.
also something to learn. But there's also something to learn from there. They were very careful about each detail, and they were just very professional. So after reading the study, I know that I have to decontaminate myself with an Alkanox Liquinox every time I enter the lab, which is of course very useful. That was a joke, by the way.
I'll get you
So what
some. We'll
now?
get you some Alkanox, man. Get you some
That's
Liquinox.
great. I found it on Amazon. That
How
should
much
be
is
great.
it? How much is it?
I forgot, but I think it's like $60 or something. Not
Is it
too
on
bad.
Prime? 2 days?
You bet. Okay, so we've all accepted it. Oxybenzone sucks. Sunscreen sucks. Hopefully, I've
I like it.
horrified you guys sufficiently. But what now? So, like, what should you do with all this information that I've dumped on you guys? What should you do now you know that oxybenzone sucks? Download my app. So, I made this app called Saga Sunscreen Advisor. And if you have an iPhone or iPad, you should download it right away. So what this app does is that it uses the phone camera and it will scan the barcode of any sunscreen. And based on its ingredients, its active ingredients of that sunscreen, it will give you a recommendation whether to use the sunscreen or not. And another pro of scanning just the barcode is that if you're ever shopping for sunscreens online, like you can go to Amazon, they'll always have a picture of a barcode. So you can just scan using my app on the computer and it will tell you whether or not to use this. And here's an example. I scanned a NOAD bar of sunscreen and it told me not to use this sunscreen. The harmful ingredients are all listed here. And it also has a list of live coral destruction from the National Oceanic and Atmospheric Administration. So the purpose of this map is just to make users feel guilty for using sunscreen in case they're ever going to use a harmful sunscreen. And it doesn't just show one area of the world. You can scroll around on the screen to look at the entire globe. And here's an example of a good sunscreen, the Neutrogena Sheer Dry Touch sunscreen. And it said to use this sunscreen because it's safe. And of course, I don't have every single sunscreen in the database of my app. Like if you scan some random sunscreen that no one knows about, it's not going to be there in my app. But the pro side is that my app uses Cloud. It uses CloudKit to, so basically when somebody scans a sunscreen that's not in the database, my app will immediately notify me and I can add that sunscreen like within a day and I can find its information and add it to the database. So that way people don't have to keep updating their app. I can just add it via CloudKit. And this screen in the middle is also really important right here. I had to make it smaller to fit the title, but hopefully you can still see it. So on the starting screen of my app where it asks you to scan the barcode, in case you don't have a sunscreen to begin with, there is a button called View Recommended Sunscreens.
That's awesome.
Well, so yeah, it has a button called View Some Recommended Sunscreens, and it will give you a list of 5 sunscreens that are environmentally safe. And the good thing about this list is that it's randomized, meaning it changes every time you click the button. So if you're in a shop and you can't find any of the items on the list, you can just refresh and it will give you a different list.
That's ethical too, huh? So you're not biasing yourself, only supporting certain brands.
Mm-hmm. And one last thing I forgot to mention about my beads is that you just have a couple of beads, right? And you're probably wondering, how am I going to get this throughout the ocean, right? Because it needs to have a much bigger impact than just a bucket of water. So I've been building this rover that's remote control and it has a camera and it can just carry my beads around with the magnet and pull out sunscreen. So this rover can go really fast. So it can just drive around and the beads can absorb the sunscreen in the water. And also my beads can be used 2 to 3 times. You just need to rinse them after use. So if you are an iPhone or iPad user, please download my app. It's really useful. It's great for the environment and it's also very easy to use. And here is the full title of the paper along with the authors. That's it.
All right, good job, man. That was awesome. I feel like I learned a lot about this paper and about your study.
I'm glad.
Yeah, do you guys have any questions for Sriram?
There was a lot of content in that presentation.
How long
is
did
the
it take you to
exercise?
conduct all the experiments?
With the beads, I've been working on them for a couple of months. So I did a lot of research online and I found a bunch of papers. And using like information from all those papers, I could make these beads. Sorry, the presentation was only supposed to be 45 minutes, but I went way over time.
I was entertained the whole time. I thought that was
Yeah, I thought it was
good.
really cool.
That's good.
I actually have a question about, because you were talking about making your rover to like get the beads out there. So like, is the rover gonna hold on to the beads the whole time, or do you like release them and then have to go find them?
No, it's gonna hold on to them the whole time.
Okay, because the whole time you were doing this, I was like, this is super cool, but how do you not lose them in the ocean? But that makes sense.
Oh yeah, that's why they're magnetic. You can just hold them with a magnet.
Gotcha.
And it's a really powerful magnetic force too. It didn't like fall off in the water or anything like that.
So once you prove that you can reduce like large concentrations of oxybenzone, how do you prove that it can reduce really, really tiny concentrations of oxybenzone? Like, how can you say that your sunscreen is going to get, what was it, like from 338 parts per billion to the no effect concentration, right? Because that's your goal, is to go from something that's bad to something where they found no effect, or where it's suggested that there's no effect. What do you think would be the process for proving that your beads can reduce it that much?
Kind of low. So I could try a lot of different samples and put in a certain amount of my beads. And using the spectrophotometer, I can see if the concentration after my beads is below the LC50 or EC50 values.
Do you think you can measure down to the LC50 values with your photospectrometer?
I'm actually not sure about that. That's a good point I haven't thought about. Because they did use a lot more advanced techniques than just a spectrophotometer. They used like the LCMS and the GCMS, and they used multiple reaction monitoring. So they did a lot of stuff there.
Yeah, so you would probably need an LCMS or GCMS to do the measurements themselves, right?
It's true. Yeah, the spectrophotometer at the lab that I'm working with, or like the lab whose equipment I used, it worked well for like the concentrations that I was testing, but the concentrations I was testing were also kind of high, considering like what you would actually see in the ocean.
Right, which is why, not putting on these harmful sunscreens is the best solution, right? For people not to purchase these bad chemicals.
Exactly. Which is why I have my app, and also why you guys should download it.
I like that app, and I'm wondering if you've been thinking about incorporating the software into other apps that, for example, tell people what kind of shampoos to buy, what kind of makeup to buy, what skin products are good, whether or not you've been talking to those people.
That's actually exactly what I've been thinking about ever since I learned that oxybenzone is found in all products from soap to shampoos to mascaras to lip balms, all types of stuff that even go on your face. So I don't want any of that stuff near my face. So I could probably extend my app to also scan other stuff too. That would be really useful.
All right. Do you know of any people that you might want to reach out to for that?
Sure, but a couple of months ago, I talked to one of my friends' Girl Scouts team about like sunscreen usage, because they do like a lot of water polo activities, and they do a lot of outdoor recreational activities too. So they probably use tons of sunscreen, and I wanted to inform them. So a couple of them downloaded my app too, but I'm still working on like, extending it to more people.
That's great. Yeah, I know that there are actually some companies out there that have apps for beauty products where they tell you which ones are good for you. So that might be something you could do, is contact the companies that make those apps and see whether or not you guys could work together.
It's a big step. That'd be very cool. Also, I put the name of my app in the chat, and the app. When you look at the app in the App Store, the developer will not be my name; it'll be my dad's. But it's the right app. Don't worry.
Okay. Was it an age thing? It can't be you because you're too young.
No, my dad didn't want to pay for another Apple ID.
What was the most interesting thing in this paper for you? What was the thing that you're like, oh wow, I didn't know that? Was it the fact that oxybenzone is in other products, or was it something else?
I think all of the transmission electron microscopy scans were pretty mind-blowing. It wasn't any one of them in particular, but just like seeing physically how horrible the effects were was really cool. Like at first I didn't really get what they were saying, but when I read the paper again and analyzed it better, I saw like I really understood the TEM scans, and it was really scary. And out of the methods, I thought that the coolest part was a trypan blue procedure with the new bar hemocytometer. So that's the one that like colors the dead cells blue and then counts it under a microscope. Thought that was really neat.
Yeah, that's really interesting. All right, we'll take maybe 3 more questions and then we'll end it.
Yeah, I have a question. What gave you the idea to use like beads? Like, did you have a specific thought in mind to just use that geometric structure?
So magnetic nanoparticles are not a new idea. They've been used in like medical fields before too, like in people's bloodstreams to do different tasks. And chitosan is also not a new thing. So I looked. There were like a lot of mixed procedures online, right? I had to find the right one because there were a bunch of papers saying one thing and then a couple of websites saying a different thing. So I had to like find the right procedure to mix the magnetic nanoparticles and chitosan. I'm not too sure where I really got the idea. I don't know. It was a while back. It just happened.
You mentioned using the beads in your robot, right?
Mhm.
Have you thought about using like a sheet to like on the bottom of your robot instead of having the beads kind of lined? I'm not too sure how you're using the beads with your robot, but when I was imagining it, I was just thinking you could probably have a sheet just on the bottom of your robot that could probably pick up more.
A sheet with like the beads attached, or
Yeah, or a sheet of the material that the beads are made with.
oh well, the part of the beads that actually absorbs sunscreen, the chitosan is pretty weak on its own, so I couldn't make a sheet of that. But maybe if I back it up with something, then it could work. But that would also probably make it a lot harder for the rover to go around if it just had a sheet on the bottom. And about the robot, like what I was thinking is that it could probably, even I haven't figured this out completely yet, but it would probably have a magnet attached with my beads or like a really thin net with the beads attached, and it would just go around and the sunscreen would attach to the beads.
Yeah, that's a super cool idea.
But I liked your idea too. It would probably be pretty effective to have a sheet there.
Sort of adding on to that, could you possibly like, I don't know, attach it to a boat or something?
A boat? Yeah,
Yeah.
yeah, you definitely could. Like that's basically what the rover is, just a mini boat. And
Oh, okay.
another idea I had in summer camp was that you could actually attach it to your dive suit.
That's a good
Like
one.
when you're diving near a reef, you could just attach it to your dive suit or like to your wrist. It'd be cool. Especially because you go very close to the reefs when you're diving. So if you can clean some sunscreen out of there, it'd be really useful. But like the beads being on a person probably would not be very effective compared to a robot because you can't carry much and you also can't cover much area. But yeah, a boat would be a good place to put it because boats, of course, travel a lot.
Yeah, like they just have it attached, just like regular commotion, like I don't know, just like any boats, like doing anything in the ocean.
Yeah,
Just thinking, yeah.
and they could also carry a lot of beads because they're so big.
All right, I have a question. If somebody else has another one, go ahead and ask it. I'm just too, I'm too involved in this. Too in it to quit it. My question is this, right? So you got corals, they're in a place like Coral Bay or Cruz Bay or whatever, they're being exposed to these contaminants all the time. Now, do you think that corals could build a tolerance to the contaminants? And do you think that this study was able to find that? If they, so if corals can build a tolerance, would this study actually show that or not?
This study didn't show anything like that, and it didn't say anything like that either. But it would kind of make sense because, I mean, all organisms adapt, right? But I think that I'm not sure at all. This is just my guess, but my guess is that BP3 might just be too bad. Like, if you feed humans cyanide, we're never going to be adapt enough to survive that. So it's kind of this. I think it's too. Could be. The study didn't say anything about that.
Yep. So there's some products that could be just so bad that you can't really build a tolerance. But in this case, like some of the St. John sites, other places, they were being exposed to these contaminants, but it could be that they could develop a tolerance to where the concentrations that they were exposed to wasn't as damaging.
That's true. And also in the experiments, they tested them in microplates. So there are no waves in the microplates. There are definitely a lot of waves in the ocean. So it'd probably be more dispersed than it was in the study. Because they didn't say anywhere that simulating the wave response.
They didn't collect that much volume either, right? It was only 500 milliliters. And, it matters where you collect the sample from, right? If you collect it from the surface waters or from, like, the deep where the corals are.
They mentioned that. They talked about that in... let me go back to my notes.
But it wasn't, like, right next to a coral, or it was?
No, it was. Some of the samples were close, and some of them were farther off. One example off the top of my head was in Trunk Bay. Wait, no, that wasn't it. One sec. Let me go back to my data table. Oh, right, yeah, no, it was Trunk Bay. So they had this large coral, and next to the coral, they found that the concentration was 1.395 ppm, which is pretty high. That's like more than most of the LC50s in light exposure. So that's really scary, but also that could be because a lot of people swim near the coral. But 93 meters to the east of the coral, they still found 580 ppb, which is still a lot and enough for like the EC50s. So they measured in a lot of places. Like they didn't measure only next to corals, but most of the samples were collected above coral remnants or current coral reefs.
Awesome. So knowing this, it actually can limit where you need to install your beads, right? So mainly you'd be focused on like high density places.
Oh yeah, definitely.
Lots of tourists.
Yeah, I wouldn't want my rover somewhere like Caneel Bay with 17 people coming there a day. Like right. Public place.
All right, awesome. That's all the questions for me. One more if anybody has a question, and then we'll we'll free Sriram.
I think it's more as a comment. I just downloaded the app actually.
Oh, that's awesome.
And I scanned a sunscreen that's not in the list. That's probably horrible. It's 100% 100% SPF.
Oh, actually, if you guys don't mind, I can talk about SPF a little bit.
Sure.
Yeah, sure.
Yeah, I recently read this article from like this Yale study that said that SPF is actually it's kind of hard to explain. So like if you have an SPF,
could you repeat that? You're cutting out a lot.
Yeah, so if you have like a sunscreen with SPF 15, that means that sunscreen lets in 1 out of every 15 UV rays. If you have a sunscreen with SPF 30, it means it lets in 1 out of every 30 UV rays. So it's like that. So after sunscreens from SPF 30 to SPF 50, there's only like a 1 or 2% increase in how much it protects you. And Yale said that pretty much all the sunscreens above 50 are pretty much bogus because they just add a bunch of chemicals and it doesn't actually increase your protection at all by even 1%. So it's like really, really tiny, and they do a lot of chemicals in there just for that tiny effect.
Could you tell us what's in your sunscreen, Ariel?
Yeah, I saw the Oxybenzone. It's 3.5%. It has two others as well. It has avobenzone and
Yeah.
octoscrylene.
Yeah, my app would tell you not to use that.
Dump
But yeah, I'm
it on a
sure.
coral reef immediately.
Yeah, but I'm sure that your sunscreen is in the cloud kit right now, so I can add that today.
Yeah, just it said it wasn't identified and said you would add it. So, yeah, it worked
Hey,
pretty
it
well.
works.
Yeah.
I can't afford an iPhone, so I'm gonna need the Android version stat.
Okay, I'll be working on it. Oh yeah, a little bit more about the SPF. So for 50, it lets in 1 out of 50 UV rays. So that means it gives you 98% protection. And for 100, it lets in 1 out of 100 UV rays, which means it gives you 99% protection. So between 50 and 100, it only increases by 1%. It's just not that effective.
Yeah, plus you won't get that tan pod, you know?
Mm-hmm.
You can't have that nice summer brown.
Apparently, going to tanning beds before age 70 increases your risk of skin cancer by like 70% or something.
All right, stop with these facts, okay? You're starting to make my future look not too good.
Full geek mode.
All right, well, thank you so much, Sriram. Here's
Thank
me
you.
clapping. Great job.
Thank you.
You are free, sir. Free to
Okay.
go build your robot nanoparticles.
Okay. I hope you guys enjoy the rest of your summer.
Yeah, you too. All right, bye.